I have written 312 biointake reports in my career. I know what the panel looks like when it returns clean data. I know the instrument signatures for a corrupted read, a contaminated sample, a probe calibration failure. I know the sound the analyzer makes when it cannot place a compound, a short, flat tone, almost apologetic, as if the machine is embarrassed by its own ignorance. I heard that tone 11 times in 40 minutes, the morning we processed the human. Her name, according to the transfer manifest, was Davin Kelso. Occupation, cargo systems engineer civilian. She had been aboard a vessel intercepted at the edge of the Venice corridor, a routine interdiction, no resistance offered, no weapons declared. She sat in the intake chair with the posture of someone waiting for a transit connection, one leg crossed over the other, hands loose in her lap, reading the room with mild, unhurried interest. She was not afraid. That, I noted at the time, was unusual. Most subjects, regardless of species, show some physiological indicator of stress during intake processing. Elevated pressure, widened pupil, an uptick in cortisol-equivalent compounds. Davin Kelso's panel showed none of it. I logged it as a behavioral note and moved on. The biointake panel itself is a 10-minute procedure. A dermal probe draws a tissue sample and a blood draw simultaneously. The analyzer identifies the subject's biochemical baseline, flags any active pathogens, catalogues known immune markers, and cross-references the result against the Cooperative species registry. For known species, it takes 8 minutes. For an unregistered species, it takes longer, because the instrument has to build the baseline from scratch rather than confirming against a prior record. For Davin Kelso, the first anomaly flag appeared at minute 4. My junior analyst, Prev Soltheis, was running the physical measurements at the secondary station. I was watching the blood panel come up on the primary display. The analyzer had identified her biochemical baseline without difficulty. Humans were in the registry. We had a reference profile, but when it reached the immune marker catalog, it stalled. The progress bar stopped at 31% and sat there. I waited. After 2 minutes, I flagged the instrument and ran a probe diagnostic. The probe returned clean. The instrument was functioning correctly. It simply had not finished cataloging. I let it run. At minute 9, the catalog reached 60%. At minute 14, it reached 73%. I called Yurin Asev, our senior pathogen analyst, over from her station. She looked at the display, looked at me, and asked if the sample had been contaminated during draw. I told her the draw had been clean. She pulled a stool up and watched the screen without speaking. The catalog reached 100% at minute 22. Neither of us said anything for a moment. The immune marker catalog is designed to identify the adaptive response records encoded in a subject's biology, the molecular memory of every pathogen an individual has encountered and survived. For most known species, the catalog runs to between 40 and 200 distinct template entries. Younger individuals have fewer. Older individuals, or those from populations with high pathogen exposure, may run as high as 300. Davin Kelso's catalog returned 4,911 distinct template entries. I asked the analyzer to confirm the count. It confirmed. I asked it to flag any entries that might represent catalog artifacts, partial reads, duplicate entries, ambiguous matches. It flagged 63. 4,848 remained after exclusion. I sat with that number for a little while. Prev had come over by then, drawn by the silence. He read the display and then looked at the human in the intake chair, who was examining a seam on her own sleeve with the focused attention of someone with nothing else to do. She was not watching us. Whatever was happening at our station was not, from her perspective, remarkable. Yurin pulled the Cooperative Registry's human reference profiles. There were 16, drawn from subjects processed at various intake points over the past 40 years. The average immune marker count across those 16 subjects was 4,318. The lowest was 3,890. The highest was 5,207. I want you to understand what those numbers mean, because when I first saw them, I did not fully understand it myself. The immune marker catalog is not simply a count of pathogens a subject has personally survived. The adaptive immune architecture in a human body does not only record individual encounters, it inherits. The templates encoded in a human's biology at birth include a partial record of every pathogen the subject's ancestral line has encountered and defeated over generations, compressed, refined, carried forward as a molecular briefing from the dead to the living. What we were reading was not Davin Kelso's immune history. It was her species' immune history, written into her body whether she had asked for it or not. 4,848 entries. Pathogens survived, catalogued, defeated, and archived in the tissue of a woman who had shown no particular interest in the fact. I asked urine what the upper range looked like for the other species in the registry. She pulled the data. The highest adaptive immune marker count recorded for any non-human species in the Cooperative registry was 612, belonging to a long-lived species from the outer Veth systems with a documented history of surviving 3 separate regional plague events. 612. Preve said quietly that it might explain the drug resistance flags on the toxicology subpanel. I had not looked at the toxicology subpanel yet. I looked at it now. The analyzer had run a standard pharmacological response model against her biochemical baseline. Of the 47 compounds in our standard sedation and compliance battery, 31 had been flagged with a projected efficacy rating below the threshold we considered clinically functional. The model estimated that her metabolic clearance rate for the flagged compounds fell outside the parameters the battery had been calibrated to address. Not because her metabolism was uniquely fast, because the compounds in question bore structural similarities to pathogens her immune architecture had already encountered and filed responses for. Her body would not simply clear the compounds, it would recognize them. I thought about that for some time. A sedative works by introducing a compound the subject's body does not know how to answer. If the body has already filed an answer, even an approximate one, even a partial structural match, the compound's effect is blunted. The more comprehensive the immune archive, the more answers already on file. The broader the archive, the narrower the window of compounds that will work cleanly. 4,848 entries in the archive. We ran a secondary draw to confirm the toxicology model. The secondary panel matched the first within a 4% margin. I notated the results, flagged the intake report for review by Cooperative Medical Division, and marked the quarantine bay for protocol reclassification pending their response. Then I walked to the intake chair and told Davin Kelso that the processing was complete and she would be escorted to holding. She thanked me, which I had not expected, and stood up without assistance. She asked, as a practical matter, whether the food in holding would suit human dietary requirements. I told her we would make appropriate arrangements. She nodded, as if that settled something minor, and waited for the escort. She did not ask why the intake had taken 22 minutes instead of 10. She did not ask what the instruments had found. It had not, I think, occurred to her that there was anything unusual to find. I filed the report that afternoon. The final line took me a while to write. I had been trying to identify what exactly disturbed me most about the results, and I kept arriving at the same place. It was not the number itself. It was the inheritance. Every human walking into this bay carries the same briefing. Not because they chose to, not because they trained for it, because every pathogen their line has ever faced and beaten was filed, compressed, and handed forward to the next generation, and the one after that, and the one after that, until it arrived here, in an unremarkable cargo engineer sitting in an intake chair and studying the seam of her sleeve. She did not know she was carrying it. That was the part I could not get past. The final line of the report read, The subject's immune architecture should not be treated as an individual characteristic. It is a species-level weapons archive, updated continuously across generations, present in every member of the population at no additional physiological cost to the individual. Recommend immediate review of all cooperative intake, containment, and compliance protocols for human subjects. The current standards were not designed with this variable in mind. I do not know if anyone in medical division has read it yet. I find I am no longer certain it matters whether they do. Davin Kelso is in holding. There are, according to the Transit Manifest, approximately 9 billion more of her out there. Every one of them carrying the same archive. Every one of them unaware that they are. That is, I think, the part that should concern us most.